Hi, Assalamualaikum everyone and a very good day. So today I would like to share a bit about my research which is on the understanding the basis of male morphotype variation in the giant freshwater prawn. So when I mentioned the word prawn, what was the first thing that came to your mind? So I bet it must be probably like a bag of prawn crackers, the one we can easily get at the convenience store. Barbecued prawns, prawn curry, prawn with cheese, all those delicious dishes. Right, so I'm actually glad to say that the prawn industry has actually spurred multi-millions of dollars globally. So when we hear the word multi-millions of dollars, it could be also billions, inshallah. So this actually makes the aquaculturists happy and also the seafood lovers pretty happy as well. So I'm actually glad to share about this handsome fella right here today. His common name is giant freshwater prawn or the udang galah, and his scientific name is Macrobrachium rosenbergi. Let's get to know more about this GFP. So the giant freshwater prawns actually is well known to most aquaculturists. It is the largest member of the Macrobrachium, which is a genus meaning big arms or big calipates. Okay, it is an important crustacean that contributes to world freshwater aquaculture production. The global freshwater prawns production has been improving over the last 30 years. It actually started with about 3,000 tons and now it's reaching about 444,000 tons. However, in Malaysia, this is the statistic of the production in Malaysia. It is somehow reducing or decreasing. So in 2010, it was about 619 tons. And then in 2019, it was only 206 tons. So this decrease in the production could be caused by the reduced fitness in a given population, or it might be as a result of breedings of genetically related individuals. This is the GFP standard culture practice not only in Malaysia but also globally. They are semi-intensive. They include or involve both sexes, which is the males and the females. Culture cycle is about 4 to 6 months, and the productivity range for 1,000 to 3,000 kilogram per hectare. There is a wide variation of individuals at harvest. Okay, so let me tell you what is the significant problem here. So the females can reach about 25 centimeters at harvest, while the males are actually larger, with probably about 32 centimeter without their calipates. So their calipates itself, or the claw itself, can grow up to about 60 centimeters. So in culture, the GFP exhibit the wide variation in adult male size at harvest. So this means it exhibit a rapid increase in size variation as they mature. Okay, so this is largely due to the large differences in the individuals themselves. Even the brothers can vary in size over a 10-fold difference. So this is largely brought about by the development of social dominance hierarchy among the males, which is due to the suppression of growth. So a significant proportion of the males individuals usually compromises the total harvestable biomass. Okay, so these are the three morphotypes of GFP. The first one, we have the BC, is the blue claw, and we have the orange claw, which is the OC, and then we have the small males. So males and females GFP, they grow differently because GFP exhibit a complex social hierarchy. So the males exhibit HIG, or known as the heterogeneous individual growth, while the females do not. So HIG is actually a limiting factor for uniform growth. Okay, the presence of size variation due to HIG is a major obstacle to profitability in the GFP culture. Adult male morphotypes, again, we have the small males. They are small, slim, and they have short claws. Their claws are relatively unpigmented and almost translucent. They are not territorial. The orange claw, you see the claw here is orange. So these are called orange claws. Their second calipate's length can be about 0.8 to 1.4 times their body size. They are not territorial as well. They have poor mating success, and they have high growth rate. While for the BC, which is the largest one, they have extremely long blue colored second calipates. If you see here, usually the calipates are twice as long as their body. They are very dominant and territorial, and they have low growth rate. So the small males will transform into the orange claw with some transition stages, and the orange claw will transform into blue claw. So once they get to the blue claw stage, it will inhibit the growth of the small males, and then the blue claw will delay the metamorphosis of the rest of the orange claw to turn into blue claw. So what farmers should do at harvest, so if you see there's a size difference here between the OC and the BC. So what farmers should do is to just take out the BC at harvest. So then leaving the OC and the SM to grow. But because it is time consuming, the farmers usually just scoop everything out of the pond with the net. So what they get at harvest is usually about 20% of BC, 30% of OC, and 50% of small males. So this is actually we call it as a heavy economic loss, because if we leave the the small males in the pond, they will actually grow into OC and also eventually the BC. So if you understand, they are all in the same pond. They are given the same resources like food and space, but the ratios at harvest are different. So this is due to the social dominance hierarchy. So understanding the factors that influences or control the progression from the small males, the orange claw, and then to the blue claw can therefore assist us to potentially maximize the total biomass. Okay, so why is this study important? It is actually to understand the molecular mechanisms that control the adult male morphotypes determination, and to develop strategies that potentially will allow us to control of this character to be manipulated. Okay, so this is my research objective. It is to identify candidate genes and markers via the transcript analysis that potentially play a role in the expression of adult male morph phenotype and determine what individual roles they play. Okay, so this is the general methodology. We did the tissue collection and then we did the RNA extraction using hepatopancreas, muscle, claw, stomach, eye stalk, and ovary and testes. And it was subjected to using the NGS, using the ion proton platform. And then we did the QC of the raw data. And then we did the de novo assembly and the Trinity. We used the Trinity software for the analysis. We did the blast comparison and annotation, mapping reads to a reference. And then we did the digital gene expression analysis. Okay, so this is the result of the analysis. Our total assembled bases was around 147 millions, and then the total number of contacts was around 30,522. So this figure shows a Venn diagram representing the number of contacts shared in common between the blue claw, orange claw, small males, and also the females. So this is the heat map generated from the microberchromosome Berger library. This actually reflects the gene expression values, whether they are upregulated or downregulated. So if we see the females, the genes are mostly upregulated, and in the orange claw, they are mostly downregulated. These are the list of potential candidate genes that could potentially influence the male morphotype in this study. So we have come up with a few search strategies. And we decided to go with the male-specific genes, female-specific genes, and pigmentations, molting, visual behavior, and stress, and also some olfactory genes that are related or associated with pheromones. Identification of genes with the primary role in morphotype determination will allow a more complex model of the pathway that controls the male morphological development to be characterized. And these data demonstrate that a number of specific genes were up and down regulated among the GFP morph, and this suggests that these genes could also be important in the cell-to-cell communication between each morph. So probably it involves in the inhibition of the process, like the inhibition of growth from the SM, and then the delays the metamorphosis from OC to BC. So these are some of the examples. So development of male morphotype is most probably a complex process. We can't actually say that the candidate genes we have identified here is involved 100% in the social dominance hierarchy in the male morphotype. But subtle changes in the gene expression during their development can impact a cascade of signaling pathway or processes that can contribute to the morphotype differentiation. So findings and candidate genes, candidate reference genes that we have identified can provide a foundation for future morphotype study. and also it can be used in hierarchical structure studies to improve the efficiency of practical genetic improvement programs in farm crustaceans, in particular the giant freshwater prawns. Okay, so that's all that I have to share with all of you. So thank you very much for your attention and staying with me throughout my presentation. Okay, Assalamualaikum. Bye.